Welcome to NPTEL online certification course on Pharmacognosy and Metabolic Engineering. This is lecture 37, where I will speak about the diversity of monoterpenoids. Let us go to the board. Okay. So, what I am going to cover here, the different types of isoprenoids and diversity of monoterpenoids, and if type permits, I will talk about the product diversification originated from monoterpene synthesis. Otherwise, I will cover this topic in the next class. So, let us now go to the board. So, what we have seen in the previous class about the different isoprenoid structures. For example, C5, the precursor of C5 compound originates from DMAPP, and basically this belongs to hemiterpenes. Okay. Hemiterpenes. Now, examples are isoprenes. So, in the previous class I have drawn the structure of isoprenes. So, like CH3, CH2, C5 compound. Now, isoprenes itself, they are emitted by the plant, and when plants are under stress, when there is heat stress, they emit it, and that actually gives protection to the plant, particularly to the membranes from oxidative damage. Now, isoprenes emission sometimes huge and depends on the particular geographical locations, they can form isoprene clouds, sometimes which is called blue haze. So, it is also observed in certain parts of the world. The isoprene emissions form the cloud with blue haze, that is called blue haze. So, such that its emission is very important for protecting the plants against different abiotic stress. Anyway, that is not our discussion here. So, next is C10, which originates from GPP, and these are called monoterpenes, and next is C15, which originated from APP, which are called sesquiterpenes. Okay, excuse me with the pen. ACSQUI. Okay. Next is C30, we can put first, that comes from, that is basically the originate from squalene. Examples are triterpenes. Then C20 originates from GGPP, examples of are diterpenes, and then C40 which originates from phytoin is a triterpenes, even tetraterpenes also. Well, so what I am going to do in the next slide is this, how these are formed. That means, how this IPP and DMAPP joins and makes GPP. So, that is my point of discussion today. Okay. What I have just mentioned about the isoprene. So, if I go back to the previous slide, okay, I can also talk about here also. So, like DMAPP, so double bond here. So, this is dimethyl allyl pyrophosphate or dimethyl allyl diphosphate. Okay. So, this by the action of isoprene synthase which removes PP plus H plus, that led to the formation of isoprene. So, which is volatile hemiterpene, which I have mentioned just before. So, together with other monoterpenes, isoprene emission is responsible for this blue haze, as I said, which is observed in distinct geographical regions of the forest during the hot weather. Now, isopentenyl pyrophosphate and dimethyl allyl pyrophosphate. Okay. So, let us go to the board. This is isopentenyl pyrophosphate. So, this by the action of isomerase can convert it into OPP. So, this is so I am not writing the full word because of the time constraint. So, this can also convert it. So, this in the next step joins with another molecule of IPP. So, leaving PP out, and forms the enzyme is geranyl transferase, the enzyme is prenyl transferase, PRENYL TRANS FERASE, prenyl transferase, and it makes So, this is the structure of GPP. So, the geranyl pyrophosphate, which is C10. So, in the next reaction, by the action of another molecule of this okay, and it makes farnesyl pyrophosphate. So, the farnesyl means wow, no, I have to make this some changes here. Okay, this is so this is the structure of farnesyl pyrophosphate, and this subsequently converted into GGPP again by joining another molecule of P, and which means this is called geranyl 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 pyrophosphate or GPP, which is again C20 compounds, and this is C15. This is called FPP. This is GPP, right. And here all these enzymes are prenyl transferase. So, here also PRE transferase, here also prenyl transferase PRDNYL, and this eventually makes. So, higher molecular prenyl phosphates are formed by head to tail addition. So, what is basically I put it here is this, that higher molecular prenyl phosphates are formed by head to tail addition of active isoprene units. So, this is all about the formation of GPP, FPP and GGPP from the basic isoprene skeleton and the enzyme activities involved in that. So, now I will give you an outline of a diversity of monoterpenoids. So, monoterpenoids, diversity of monoterpenoids. So, monoterpenoids originate from the GPP. Monoterpenoids are found inside the plastid. So, this is the site of monoterpenoid synthesis and accumulation in general. Sometimes these monoterpenoids may move into the vacuole and stay there, but normally this is generally the site. So, when this is the site of synthesis, that means the reaction, that what is the precursor of monoterpenoids is GPP, and GPP by the action of enzyme which is called monoterpene synthase, is a generic name. So, specific names will come and it forms different monoterpenes. I use the term monoterpenoids because this monoterpenes having functional group attached to it. So, these are mostly oxygenated. So, that is why we will use the term monoterpenoids. So, monoterpenoids, I write it clearly MONOTERPENOIDS. So, monoterpenoids accumulate inside the plastid, that is a thumb rule, and GPP is the precursor. So, we will talk about the enzyme catalytic mechanisms later. So, before that let me show you some of the diverse structures of monoterpenes. Now, these monoterpenoids can be classified into different types. So, maybe acyclic or this cyclic. So, acyclic monoterpenoids, if I start giving you example, acyclic, ACYCL, I see acyclic monoterpenoids or monoterpenes. In general, you can a few structures may not contain oxygenated groups. So, that we cannot in true sense call monoterpenoids, we can call monoterpene, but anyway. So, loosely we are calling all under acyclic monoterpenoids. Acyclic name itself indicates that there is no cyclization. That means, say if I draw this structure, problem with the pen. So, this is the structure of medicine. This is the structure of nerol. Now, this is the structure of geraniol. Okay, now this one will not be there. This is the okay. This is structure of citronellol, and much gap পড়ে যাচ্ছে, and this is the structure of linalol. Now, so here actually there will be CH3, there will be CH3, there will be CH3. Otherwise, we if you wish, we do not also put it. So, these are the examples of acyclic monoterpenes. Okay. So, medicine, nerol, geraniol, citronellol, linalool all contributing to the flavor and fragrance from different plants, including tea, including different flowers, rose scented geranium, rose itself, so many plants. Okay. Specific examples will come when I will when I will talk about specific cases. Now, I will give example of cyclohexanoid monoterpenes or cyclic monoterpenes for simplicity. Cyclic monoterpenoids. This is basically the example of limonene. Limonene you will find it in the lemon. Okay. So, the lemon you have a thick peel and there lies several oil glands. So, if you squeeze it, that whatever watery droplets come out. So, that is basically that contains limonene along with other metabolites, other terpenes. So, limonene is the signature molecule of this rutaceae. Now, another example, let us say carbon. So, this is carbon. Another one, say menthol. Now, this is 1, 2, 3, 4, 5, 6. So, in case of carbon, this is at 6 position, 6 position get oxygenated, whereas in case of menthol it is 3 position. So, the precursor molecule limonene undergoes different enzymatic reaction, depends on the different plant. So, menthol you find it the peppermint, PIPPER, peppermint, or PIPER, peppermint. Carbon you find it in pudina, that is spearmint. Pipermint, okay, spearmint, okay. Limonene you find in the lemon. Similarly, there are other compounds, for example, pulegon. Pulegon is basically a precursor of the menthol. Pulegon okay, and then menthol from menthol, actually menthol is formed. Sorry. The other examples are carviol or isopipe. Okay, I will put another important example, which is cineol. This is called 1,8-cineol or eucalyptol. Similarly, another compound is called phelendrin. And I can also put example of camphor. And pinene, which you find in the pine. There are two types of pinene, one is called alpha pinene, one is called beta pinene. this is alpha pinene, and here here CH2 beta pinene. Another example is carviol, right. So, this is more or less okay. Two more things I will draw and finish this class. One is the thymol, and another is carvacrol. So, you see that the product diversity of different cyclic monoterpenoids. So, they all originate from GPP, and through different through changes in the reaction mechanism leading to formation of different monoterpenoids. And as I said that monoterpenoids is a very versatile enzyme, monoterpene synthase, and there are specific monoterpene synthase which actually gives one product formation, whereas there are other monoterpene synthase which can be as many as 8 monoterpenoids catalyzed by a single reaction. From GPP, monoterpene synthase gives rise to 8 different monoterpenoids. So, in the next class we will talk about that, and we will talk little bit of this type of skeletons, and then we will go to the catalytic mechanism for the formation of these monoterpenoids from the GPP, which is itself a bit complicated, but I try to put it in a simplified way, so that you can understand. And the point is this, that beta phenyl. What is this? Okay, I have given you the example of this, like thymol you get it in thyme, okay, carvacrol you will find it in Lamiaceae species, carviol also, pinene you find it in the conifers, cineol, eucalyptol you will find it in several plants including Nicotiana species as well as tuberose, so many, camphor you find in the camphor plant itself, like so many things. Pulegon and menthon you find in the peppermint. So, the diversity is enormous, and accordingly the enzymatic reaction is also very unique, or rather more diverse towards the formation of different products. So, with this I end the class. Before I end the class, let me see what I intend to cover. So, different type of isoprenoids I covered, diversity of monoterpenoids I have covered, but product diversification originated from monoterpene synthase, that I will cover in the next class. So, with this I end the class here. Thank you.